If you asked some geneticist 25 years ago whether we would be doing the things that we're doing today, even whether we would know the sequence of the entire human genome, they would have said, no way that's going to happen in that period of time. This is really big stuff.
People see cloning as a way to raise the dead.
Cloning, as it's most customarily practiced at the present, involves the transfer of nuclei from cells and transferring those cells into eggs where the nucleus has been removed, and then the nucleus from the animal to be cloned is put in its place. If it's a mammal, those new embryos then have to be transferred into recipient females for them to carry on to term, and usually those recipient animals nurse them and raise them as well.
K9. Cloning techniques. Give me a rundown, state of the art so far.
Cloning is replication, making a copy of an individual from a single cell of that individual. Clones retain characteristics of original organism.
Let me show you what it looks like. So you can see here sheep eggs, a large pipette on the left-hand side, which is going to be used to pick up the eggs, and a smaller, sharp-tipped pipette on the right-hand side, which is going to make the holes in the outer shells, which you can see, translucent outer shells, and is going to be used to remove the genetic information. So he's picked up one of the eggs, going to look at it under higher magnification. Now, one of the problems is you can't see the chromosomes. But in a minute, you'll see a small sphere of cytoplasm at 6 o'clock. And we've every reason to think that the chromosomes are very close to it. And so he's going to remove it and some of the nearby cytoplasm. In order to know whether or not he's successfully removed the chromosomes, this egg has been incubated in a dye which binds to DNA and fluoresces in UV light. So he takes the egg out of the field of view, turns on the light, and there are two fluorescent bodies. One is the chromosomes that were in that little sphere of cytoplasm, and the other is the chromosomes that were in the egg. So this time he's been successful. If he didn't see two groups, then he'd either have to discard the egg or try again. So he then has to provide the new genetic information which is going to control development. In the case of Dolly, these cells were grown from mammary gland of a ewe that was about 2/3 of the way through pregnancy. He's picking up several of these cells. And since this is the same pipette, you can see that these are very much smaller than the egg. And what he's going to do is to go back to an egg from which the genetic information has been removed, and he's going to put that cell in the space between that outer shell and the big cell in the middle. And we're then going to use an electric current to do two things: to fuse the two cells together and to stimulate the egg to begin development. The electric current will only work if the two cells are touching, so the last thing he'll do is just to push them together. We'll just watch him do that one more time. During a fairly intense day's work, he would be able to attempt the process with something like 100, 150 eggs. There will be a technical problem or a biological problem with about half of them. So a really good day's work would be to have 60, 70 eggs, new embryos made in that way. So then what we do is to put those embryos into foster mothers, surrogate mothers, which will give the embryo the chance to develop to term. And sometimes we would have a lamb which would have the opportunity to grow up into a healthy adult. You may remember that Dolly was just one lamb from more than 270 attempts. The efficiency's increased just a little bit since then, but it's still of the order of 2 or 3%. With an adult cell in sheep.
There will be continued research to make it more dependable. At present, about 25% of offspring have some type of abnormality. They're developmental abnormalities. They're not mutations. They don't get passed on to the next generation, but they need special care.
The truth is that cloning is highly experimental and very problematic, and most of the time it doesn't work. Only a few clones have actually been born successfully, And of the ones that have been, a good percentage actually end up dying within the first 6 months because they have some sort of complication, health abnormality, a physical deformity, something like that. You talk about using a very large number of animals to even try to produce just one clone.
An adult would be cloned would be you have to take a cell from that adult, and you might be able to get that from a tissue swab in the cheek or from a scraping of skin or from a little biopsy, and the cell would then be taken to the laboratory.
Once they have selected the cell, scientists will take its DNA and insert it into the empty egg. Then they will have to stimulate the egg to begin to grow. They might use an electric shock and add chemicals, the methods used in animals. No one understands how or why, but this process should switch the adult cell back to become the very first cell of life. The new embryo will be grown in a dish for a few days, then implanted into a woman's womb, where it might grow into a human being. This human would be a clone. And away from the hype about cloned babies, mainstream scientists do also see good reason for cloning from human cells. They're hoping to treat incurable conditions like Parkinson's or heart disease by growing new brain or heart cells cloned from the patient. So is science really ready to clone a human?